So here we see the syringe needle going in and the lipid nanoparticles coming out. Probably 3 to 6,000 billion per injection. 3 to 6 trillion. And the body's never seen lipid nanoparticles before you had your first mRNA vaccination. These are completely new to the human body. And they circulate around in the blood. Here we see it going through a blood vessel, circulating around with the much larger erythrocytes, the red blood cells. And the body's never seen these lipid nanoparticles before. And this is before we even think about what they contain. Nano means very, very small. And they'll go everywhere. They're supposed to stay in the deltoid muscle, but they don't. They circulate everywhere around the body, even if they're not given via an inadvertent intravascular injection. They're going to go to all 5 toes. They're going to go to your ears. They're going to go to your heart, your liver, your spleen, your kidneys. These things go everywhere, the ovaries and the testes. Then when they come to a cell, they'll go inside the cell, this process called endocytosis. And this modified foreign RNA will end up inside the cell, as will the lipid nanoparticle itself, into the wall of the vascular endothelium, the linings of the blood vessels. And because the RNA is in this lipid nanoparticle, it avoids the immune system. So will contaminating DNA avoid the immune system and be transported straight into our cells. Any contaminating DNA will be carried very, very efficiently by the lipid nanoparticles into cells. And we know they go everywhere because this one's been labelled with luciferase, which gives off light. And these things can be seen in all organs of the body because they circulate everywhere in the blood, as we see in this amazing graphic, coming into contact with the walls of the vascular endothelium, the lining of the blood vessels. So this diagram gives us another sense of scale of the lipid nanoparticles. Here's the lipid nanoparticle here, with its fatty membrane outside. Here's the modified RNA inside, and this is actually a SARS coronavirus too. Now remember that viruses themselves are too small to be seen through a light microscope. We need to see them through an electron microscope, and even then we don't get a great deal of detail. So we see that the lipid nanoparticle is way smaller than the size of a virus. So it's no wonder these little things can get everywhere, because they can go through very small gaps indeed. Now this massive structure we see here is a red blood cell. So this is all one red blood cell. These are about 7 micrometers in diameter across. And if we get lipid nanoparticles, if we assume the lipid nanoparticles are 80 nanometers each, which is about right, then we see that it would be 87 of them to fit across the diameter of a red blood cell. And bear in mind, these red blood cells are so small, there's about 5 billion, 5,000 million of them inside 1 milliliter of blood. And on that scale, these are the lipid nanoparticles. In fact, that would be quite a big one that I'm drawing there. This is how small they are compared to red blood cells. So 5 billion of these red blood cells in 1 milliliter of blood, and yet this is the size of the lipid nanoparticles. It's no wonder they can get just about everywhere in the body. Now, those graphics just really bring the situation to life, don't they? They were provided by Erik Tangsoo. These are his links. I've put them at the top of the description. He's not being paid for this work. Please click on his videos and encourage his work with comments and share them around. very much more detail in those videos. But that just gives you a bit of an insight into these pesky lipid nanoparticles. Until these problems are resolved, there should be a moratorium on mRNA vaccines. In my view, let me know what you think. Now in this next group of graphics from Archie, we're going to look at the modified ribonucleic acid and how it's been changed and the harmful effects that this can potentially have. So here we see a strand of RNA, the ribonucleic acid. But the problem is it's not ribonucleic acid, it's modified ribonucleic acid. It's been modified to avoid the immune system and to last much longer. Ribonucleic acid has been around as long as humans have, ever since, well, longer than humans have, as long as life has on the planet. You know, the simplest bacterial cells contain this wonderful God-made natural RNA, but this is a synthetic form. This is completely different. We're dealing with a different phenomena here, human engineered. It's been changed in various ways that concern us. And in particular, what we have in RNA is we have bases. So in RNA, it's normal to have these bases. There's adenine, uracil, cytosine, and guanine, A, U, C, and G. But the uracil has been switched out for pseudouridine. So pseudouridine has replaced the normal uracil. And we can see these as these three spiked prongs in this rather excellent graphic. They've been replaced by pseudouridine. So this pseudouridine, this false synthesized uridine, engineered to last longer, it's actually based on genetic sequences only. It's basically generated by a computer and the bases are strung together. And it's read by our ribosomes over and over again. So natural, brilliant messenger RNA that's been designed to transfer the genetic code as long as there's been life on the planet Earth has been made into this synthetic, modified form of RNA. David Speaker actually described it as being like a plasticky thing that just persists in the cell. And in fact, we know from some studies that it's been detected 700 days after injection in post-vaccine syndrome. We see all that the natural uracil's been taken out and it's been replaced with the pseudouridine. And this can cause quite a few problems. It can cause misreading of the natural sequence because of the abnormal pseudouridine. The function of the RNA can be changed. This rather brilliant graphic here is showing the way that the RNA goes through the ribosomes being the cell's protein producing mini factories that translate the genetic information in the RNA via the ribosome into, should be the body's own protein, but of course in this case we see that it's making a spike protein instead. And as we've said, because there's a pseudouridine in there, the RNA can be misread, can be frame shifted to produce abnormal proteins, in addition to what you might call the abnormal spike protein. Yeah, one study's shown there's about a 10% error rate. So maybe 90% of the time this modified unnatural RNA is producing spike protein. The other 10% of the time it's producing proteins that are not spike protein, but not natural human proteins either. Inside the lipid nanoparticle, the spike is protected from the body's immune system. The mRNA strand is inside the lipid nanoparticle. Injected directly into the systemic spaces of the body. So when it's time to make some more protein, part of the DNA in the nucleus of the cell will unwind. And an enzyme called polymerase II will go along the DNA. And it will take that information from the DNA and translate that into mRNA, messenger RNA. And this messenger RNA will then trot off into the cytoplasm of the cell. It's actually guided there via quite precise mechanisms, but it goes off into the cytoplasm of the cell. And in the cytoplasm of the cell, we have the ribosomes. So the information is transcribed from DNA to RNA. The RNA gets to the ribosomes where that information is translated into proteins. And that's how the body makes its own proteins. So I like to think of it as DNA is the recipe that you print off your computer or read in your recipe book. Then I go into the kitchen and I want to actually make the meal that the recipe codes for. And it's me that puts it together. So in a sense, I am the messenger RNA or the ribosome that's putting this meal together. Rather appalling analogy, but maybe that gives you the idea. The DNA is the information. It's then put together, operationalized into the essential proteins that keep us alive. And actually, from time to time in DNA, there are mistakes called mutations. And these are remarkably useful sometimes because they can help us to adapt into different environments. So, for example, if we go and live up a mountain, we can make hemoglobin, which is more suited to low partial pressures of oxygen. But let's have a look at a brilliant graphic that shows this actual process of the RNA being produced in the nucleus of the cell. So in the chromosomes, the DNA is tightly packed. But because we want to make some proteins based on the information in the DNA, here it's been unwound in the nucleus of the cell. And here we have the polymerase II that's going to go along, read the information from the DNA, convert that information from the DNA into the messenger RNA. This is what is supposed to happen. Let's just watch this happening now. And there we see the RNA molecule carrying the information from the DNA molecule. This is a mirror image, really, of the information that's in the DNA, ready to take this information out to the cytoplasm, containing the information that codes for the immensely complicated body proteins. The mRNA is then transported with its message. It's mRNA, it is messenger RNA carrying this complex genetic information for the production of cellular proteins, which of course is essentially what most of the body is made from. We are largely made from proteins. And the analogy used in this graphic is that of the DNA being the instructions, the blueprint, and then the project managers being the RNA bringing it to life, which is fine. So you can have that analogy, or you can have my recipe analogy. Take your pick. Well, welcome to this talk. In this talk, we're going to look at how the vaccine mRNA basically cons our cells into producing spike protein, how that spike protein goes onto the surface of our body cells, potentially anywhere in the body, could be the heart, lungs, brain, ovaries, kidneys, testes, and how that spike protein labels the cell for destruction by our own immune system. And our own immune system is very good at that. It will destroy our own body cells in autoimmune pathology. Well, here we see the SARS coronavirus 2 viral particles, and we can see the spike protein that's expressed from the surface of the virus. This is what allows them to infect human cells. And the spike protein can also be released into the blood from the vaccine. But here, what we're actually seeing in this graphic here is we're seeing the ribosome. This is the mini protein producing factory inside our cells. And here we're seeing it reading this RNA here, but this is the synthetic RNA from the virus. So in a sense, this is producing a synthetic spike protein. But it's being produced, the spike protein is being produced in the cytoplasm of the cell as the ribosome translates the genetic information from this genetic sequence that's been injected and transported via the lipid nanoparticle to produce this spike protein. In a sense, we could call it a synthetic spike protein. And this RNA strand can be read many times, giving a very unpredictable dose. No one knows how long it's going to carry on producing spike protein for, although in some people it's a long time. Now here's the spike proteins here, growing up. But here they're growing up on the surface of our own body cells. So these look like mushrooms popping up here, but it's not. This is the spike protein being produced, popping up on the surface of body cells. The spike protein that's been produced by this synthetic RNA that's got there from the lipid nanoparticle. And because the lipid nanoparticles are systemically distributed, because they go everywhere, this can happen in any cell of the body. Any cell of the body that the messenger RNA comes into contact with can start producing these spikes. And of course, these are quite abnormal. They're not supposed to be there. They're foreign proteins. Now, what's supposed to happen when a cell is infected by a virus? And remember, viruses are obligate intracellular parasites. They can only reproduce inside other cells. And the immune system patrols the outside of the cell. So the immune system patrols the body humours and the body tissues. It doesn't go inside the cells. So if there's a viral infection inside one of our cells, that viral biochemistry is going on inside the cell. And the cell wants to alert the immune system that there's a viral infection inside it. But the immune system can't see into the cell. So what the cell does is very clever. It takes some of the viral proteins and it transports those to the surface of the cell and it expresses those on the body cell. So if you have a virally infected cell, that will be expressing viral proteins on the surface of your body cell. And in that case, the immune system will see it and the cells of the immune system will destroy that infected cell. So to begin with, we have cells called natural killer cells, the large lymphocytes, and they will kill virally infected cells. And then later on we have cytotoxic small T cells, small lymphocytes, that will kill virally infected cells. Now the reason that the body kills the virally infected cells, even though it means killing its own body cells, is that it's killing the places where the viruses are replicating. If the immune system doesn't kill the infected cell, then that cell will generate thousands of copies of the virus. They will float off and infect other cells and the viral infection will proliferate. So the body thinks, so let's cut my losses and we'll just eradicate the cell. So here we see these popping up. These spike proteins are now popping up on the body surface cells where they're not supposed to. Very often these are probably in the vascular endothelium, inside the blood vessels. And the cells lining the blood vessels, the vascular endothelium. And this could be in any blood vessel anywhere, in the heart or the kidneys or the brain or the testes or the ovaries. It could be absolutely anywhere that the spike protein goes. So remember, the cells, when they're infected by a virus, they want to say to the immune system, hey there, I'm here, come and destroy me. You know, I'm a source of viruses, you need to get rid of me. And it does that by labeling viral proteins on the cell surface. And that's exactly what's happened in this situation. And I think you probably get the next bit of the story now. Now it's labelled with spike protein, the immune system will come along and destroy that cell. So here we see spike protein in lots of body cells. Spike protein expressed by many body cells in the vascular endothelium lining the blood vessels. And then antibodies are going to stick onto the surface of these spike proteins. And what happens here is these antibodies act as something called opsonins. They label that cell. So the antibodies will get between the spike protein and the immune cells. They'll communicate between those two, and the immune cell will then destroy the body cell. And that's exactly what we see happening here. We see that body cells here and here have been destroyed, leaving gaps, basically leaving wounds in the surface of the vascular endothelium, because the body has been conned into thinking that these cells are infected with a virus because their surfaces are expressing viral proteins. And here we see lots of cells in the vascular endothelium dying because they're being killed by the body's own immune system. Now, this is bad, and it's bad for several reasons. First of all, well, it's a wound, isn't it? So that's bad. It's an inflammation. But as well as that, this vascular endothelium normally produces anticoagulants that keep the blood liquid, because the blood in circulation is very liquid. It has to be liquid, so it circulates around everywhere. So the damaged surface endothelial cells, these cells here, are no longer producing this anticoagulant for the surface. So we've got reduced anticoagulation on the surface, making coagulation, that's blood clotting, more likely. Also, now we have wounds. Now, when the platelets in the blood, the thrombocytes, when they come across a wound, like if you've cut your hand, they come across a wound and they think, there's a wound there, we better start clotting together and initiate the coagulation cascade to form a blood clot, otherwise our person is going to bleed to death. So obviously, when the platelets come across a wound, they initiate blood clotting. That's why we don't all bleed to death every time we get a little injury or a cut. But of course now, the wound is inside the blood vessel. So that's going to stimulate blood clotting or thrombosis as well. So we've got the lack of the natural anticoagulants that the vascular endothelium is producing. We've got the platelet activation as a result of the wound. And as well as that, spike protein can initiate clotting directly. So we've got a triple whammy that can cause blood clots. So that's not good, because we are essentially wounding the inside of the vascular endothelium by the body's own immune system, as the body's own immune system recognises the spike as being foreign, thinking it's a virally infected cell, and therefore correctly obliterates our own body cells via autoimmune processes. So the vaccines are just producing the spike. And of course, it's the spike proteins that are the toxic part of the virus. So why the manufacturers chose to produce the most pathogenic part of the virus is rather strange. Why didn't they choose to stimulate the immune system with a more benign part of the virus? It's very strange. I don't understand that one. But anyway, that's what they did. Anyway, let's watch the rest of this excellent graphic from Arki. Links below. Do click on his videos. Yeah, so the vaccines were based just on the genetic code supplied, hopefully early on in the pandemic, by the Chinese. Now here we see the blood clot. We see that blood clot there. So as a result of lack of anticoagulants produced by the vascular endothelium, as a result of platelet activation caused by activation of the platelets, initiating thrombus formation or blood clot formation, and as a result of the spike protein itself, here we've got a blood clot inside a blood vessel. Blood clots on the surface of the body where they're preventing bleeding to death, brilliant. Blood clots inside blood vessels, very bad. That's called a thrombus or thrombosis. So a thrombus is the formation of a blood clot inside a blood vessel, which should never have blood clots. And if bits of that break off, that's called embolism. Emboli can break off and fly to different parts of the body, potentially blocking off bits of the circulatory system. So here we see that a blood clot is formed. We see that there's red cells in it. We see that there's white cells in it. And we see that it's held together by these fibrin strands. These fibrin strands are supposed to be in solution. What happens is you have fibrinogen in the blood, but when the coagulation cascade is triggered, this fibrinogen is converted to fibrin. So fibrinogen is in solution, it's liquid. Fibrin is a long sticky strand. Some people call it fibrin. Fibrin, exactly the same thing. And the blood clot, the blood cells will stick to that, causing the blood clot where we don't want it, which is a thrombosis. We see the spike protein causing all sorts of trouble, both by direct toxicity and via autoimmune mechanisms. And here we see the antibodies that the spike protein causes to be generated. Well, I think you'll agree they're pretty amazing graphics from Arki Tangsoo. Now, he's made these available freely. Do click on the links below to watch the full links of his films. They last just over an hour each. Really quite superb. One on the vaccines, one on the vaccine trials. And yeah, support him any way you can, because he doesn't get paid for this work. So thank you very much for allowing us to use these graphics. And of course, thank you for watching.